Hi, I am Mrs. Sloan, and this video is intended for my AP Biology students, and this is Immune System Part 2. So what we have already gone over, here, let me make myself a little bit smaller, is we have already discussed the evolution of the immune system. So if you want to have a little background on that, I suggest you look at video 1. We have talked about PAMPs and toll-like receptors. We have talked about how it relates to signal transduction, and we have done an overview of the innate immunity, what you're born with, versus adaptive immunity, which is what this video is about adaptive immunity. We have also talked about the role of the immune system, your lymphatic system, and when things go wrong. And we've talked about the important organs, and so you want to be familiar with that. And then also all of the defenses we have in order to fight off invaders using our innate response. So now, and also about cytokines and complement proteins. So now what I want to do is talk to you about adaptive immunity. And let me present. And I'll show up somewhere. There we go. And what do you hear in adaptive immunity? Adapt, right? So it's changing to the environment that you're in. And that's one way to maintain homeostasis, right? So the big difference is your innate immunity. These cells can respond to any invader. They can tell self from non-self, and basically they just attack without precision anything that's not your own body. What adaptive immunity can do is you have a finite number of cells, like in the millions though, of what you call virgin B and virgin T cells that are just waiting for some infection that's going to happen next week, 2 weeks, 10 years, or 20 years. And once that cell who can recognize that specific pathogen has been triggered, it's like a lock with a key, and that pathogen is the key, and it opens the door, and you then make millions of cells that can identify that particular pathogen. And that's why it's so effective, is you raise clone armies that can address that particular thing that's invading your body and attack it with precision until it's completely eliminated and it's extinct in your body. And then it has the ability to remember that. That's the adaptive part. It can guide your body. So the antibodies that are in my body as a result of my adaptive immunity are going to be different than yours because of your experiences in your life and what antigens you have faced in your lifetime and are still yet to face. All right, so let me get on to this next slide. So, oh, look, I'm a basophil. All right, so remember, I kind of categorize your innate immunity as your police force. They can recognize invaders. They can deal with it. Like, remember, the mast cells release histamines that makes vasodilation of your capillaries so that these other cells can get in and out. They all play different roles. And these two overlap with each other. They can help each other out. Remember, this innate immunity, anything foreign, it recognizes and attacks and tries to destroy. But over here, your B and T cells are precise. There's only a specific pathogen that they can attack and destroy. And so what you need to build is millions of each of these so they can do that job. Now, remember, all your blood cells come from your bone marrow, right? All your blood cells come from your bone marrow. And then you have your myeloid stem cells, which makes things like red blood cells and what will eventually be platelets, right? And then also this comes your innate immune response. And remember, inflammation and all of that. That comes from your myeloid stem cell. From your lymphoid stem cells, you have your special forces, your T lymphocytes and your B lymphocytes. This is the immunity that lasts years and years and years, very specific immunity that lasts years and years, and hopefully your lifetime, right? So what triggers these immune responses? If you remember, it's antigens that trigger the immune responses. These are surface markers, proteins, or maybe a carb of some sort. And remember the antigens in Winnie Sloan's body, right? I'm not going to recognize, unless I have an autoimmune disease, I'm not going to attack any of my body cells. But if you needed my kidney and we were a match and I gave you my kidney, all of a sudden, every cell on Winnie Sloan's kidney says Winnie Sloan, right, by her antigens. So when that's in your body, you're like, stranger danger, and you will attack and destroy it. So what's an antigen for one is not an antigen for another. Now, what you and I have in common is we do not like bacteria, if they're bad bacteria. We do not like fungi that are invading our body, and so we will work to attack and destroy those. We won't like viruses, so that part is the same. So on your notes, and if you are new to my classroom, in the descriptor, if this is on YouTube that you're watching this, in the descriptor down at the bottom, there is a link to the group shared notes that my students use. Column 1 is a scaffolding that you can fill in to keep track of the important items. And then column 2, add in pictures and images that are helpful for you. So on these notes, we are on 33.4. And so this is the adaptive immune response. And you want to have on there that it is also called acquired immunity because we are not born with it. Let me jump back one slide. It is called acquired immunity because we are not born with it. Adaptive immune defenses can distinguish between self and non-self. So can these guys too. Just want to throw that out there. It can take a week, maybe 7 to 10 days, maybe longer, but usually somewhere in there for it to develop, but it lasts a lifetime for years. It primarily depends on the B and T cells, and you can see those right here. And we'll talk, I have some more slides for you on that, some B and T cells that have specific antigen receptors. Remember when we talked about PAMPs and toll-like receptors, right? The PAMP or the antigen is the signal, and then the receptor for that, there are B cell receptors and T cell receptors. We'll talk about that. And then the diversification, you literally have millions of virgin B and T cells that have never been stimulated by an antigen yet, because you need to be pre-adapted for all antigens you may be exposed to in your lifetime. Maybe in 10 years you travel to Italy and you're exposed to some antigens there. You will have, hopefully, B and T cells that can recognize that antigen and respond to it and build a clone army to take care of that pathogen at that time. How do you have all that diversity? It's called exon recombination. Now, in my class, we haven't got to the detailed structure of DNA replication and transcription and translation, but we will learn about that later. So for right now, I just want you to have on your notes, diversification of antigen receptors happens during maturation process, creating virgin B and T cells that can respond to any possible antigen, through genetic recombination in post-transcriptional processes. That means you've used your DNA to make mRNA and then you modify the message, okay? Exon recombination, and get this exon recombinations for this millions of variety, you know how many genes it comes from? Three genes. So this whole response is triggered by antigens. So they are defined as any foreign substance that triggers an immune response. They are found on the surfaces of viruses, bacterial, and eukaryotic cells, and they are made of carbohydrates or proteins. All right? Now, this is a chart just to help you differentiate between B and T cells and how they function. And then I'm going to go into some details about just the antibody-mediated and then just about the cell-mediated immune response, all right? So first of all, the types of immune cells. For antibody-mediated, it's a B cell. And remember, they're called B cells because they mature in the bone marrow. And for the cell-mediated, they are called T cells. Remember, this is underneath the umbrella of adaptive immunity, right? Learned immunity, okay? So T cells mature in the thymus, B cells mature in the bone marrow, though both originate from the bone marrow. All right, its target. B cells' target is your free-floating viruses and bacteria. They are not hiding in your body cells. They are moving through your interstitial fluid, your tissue fluid, or they are in your blood, or they are in your lymph. It is the actual virus or bacterium that is moving around. The target for T cells are your own body cells that have been infiltrated by a virus or some other pathogen, and could now be harboring that virus, and thousands, millions more viruses could come as a result of taking over your host cell machinery and generating more of themselves. Or a target cell could also be one of your cells that has gone bad. Remember, we talked about cell controls and cancer and the process of losing control of the cell cycle. So if you have a cancerous cell, that will also be attacked by your T cells. And the process of that, B cells make what's called antibodies. Antibodies look like Y-shaped structures, these proteins, and I'll show you some more pictures of those in just a minute. And when they are released, antibodies can flood your entire body, okay? And what they will do is these antibodies, remember, this is all about specificity. These antibodies, you have different antibodies for every single pathogen you've ever had in your body. Those antibodies will dock with that, like you can see right down here with the bacterium, and they, remember, mark it for destruction, for phagocytosis. They also inactivate this bacterium, or if it was a virus, by binding on to its antigens. It's not going to gain access to your cell. So it inactivates it and targets it for destruction. These are your free-floating viruses and bacteria moving through your body. Conversely, your cell-mediated, with your T cells, they're going to do cell-to-cell combat. This green guy right here, imagine him as a T cell fighting one of your own body cells. And he's fighting one of your own body cells because it's harboring this virus right here. And if it doesn't kill it, then it's possible that all of these viruses will be released to your body. So it's got to take out our body cells that are infected. So that's the big picture overview. And then we'll start in on your antibody-mediated. All right, so let me tell you the story of the antibody-mediated. First of all, you have what's called antigen-presenting cells. Do you remember when I talked to you about dendritic cells, right, that eat up all the dead stuff of the battles, right? And we talked about neutrophils, we talked about eosinophils, macrophages. Remember, dendritic cells, this is part of your police force, right? And if there's a bad guy, he's going to eat this bad guy. Now, he'll eat any bad guy that there is. Anything that's not your own body, he'll eat it, okay? He will then rip it apart and then display a small portion of it on the surface of its body in order to try to elicit your B and T cells, specifically T cells, okay? So it will present part of it. That's why it's called an antigen-presenting cell. I don't especially like in this cartoon where it's presenting it, but we'll just move past that. So it presents this antigen, which looks like a red gumdrop of some sort, and it's not the whole pathogen, it's just a piece of that pathogen. And here it has docked with a helper T cell. This helper T cell would only bind to this particular antigen, okay? If it was any other shape, any other color, however you want to think of it, structurally, if it was different, this particular helper T cell could not bind to it. It's very specific to this antigen. Do you remember when we talked about PAMPs and toll-like receptors? Okay, very, very specific, this B cell receptor. Now, this T cell is activated. The light is green for him. He has said, I have finally met the antigen that I have waited my whole life for. So what happens as a result of that? Well, put a pin in that. I'll be back. Now let's look at the B cell. Notice how the B cell is stimulated. The B cell is not stimulated by an antigen-presenting cell. It goes directly to the source, right? To the bacteria, to the virus. And this particular B cell, there are 3 pathogens right here, but only one of them, the red gumdrop bacteria, is the one that binds to it. And when it binds to it, if it meets up with the virgin T cell who was activated right here, right? Remember the virgin T cell that was activated? These two have to agree. This T cell and this B cell, who have never before experienced the red gumdrop bacteria, have to agree that, yes, this is a bad guy. So this is kind of a checks and balances in your body. And so now that this B cell has been activated, okay, now what it becomes, this B cell, right, this virgin B, starts doing mitosis and forming what's called plasma cells. Now, plasma cells are going to secrete antibodies into the blood plasma, which are going to get pushed to everywhere in the body, even in the interstitial fluid, the tissue fluid, in the lymph. They will go everywhere, these antibodies. And these antibodies will even cross the placenta barrier between a mother and her when a baby's developing inside her uterus. It will cross that. So these antibodies go everywhere. And so they will make all of these antibodies which will target and land on and inactivate the bacteria. The second type of cell that your stimulated once was virgin, no longer virgin cell is a memory cell. And that is for the next time you meet this invader. So what do these antibodies do? These antibodies tag the bacterium for so it will get phagocytized, right? Another name for these are opsonins. Anything that triggers phagocytosis is an opsonin. It could be an antibody. It could be a complement protein. Could do the same thing, right? But what it's going to do now is it's going to eat them, right? And there's all the free floating ones, if they come in contact with an antibody, they're going to get tagged and marked for destruction. Now, once the battle is over, okay, you will have made millions of plasma cells secreting these antibodies. Once the battle is over, all of them will commit apoptosis, cellular suicide, because you cannot maintain for every illness you've ever had. You can't keep a whole army for every illness. You don't need them anymore. You wiped it out. You just keep a few behind, and those are called your memory cells. And these memory cells are non-virgin B cells, and they are stimulated much quicker. And if they come in contact with that pathogen, the light is green right away. They don't need to wait for a T cell to give them the okay approval. They don't need any of that anymore. They can immediately start making more plasma cells and more antibodies to take care of it. So on your B cells and antibody-mediated immunity, I want you to have the targets are free-floating viruses, bacteria, or toxins. B cells have B cell receptors that are usually activated in a lymph node. Or do you remember the other thing that filters your blood? It's the spleen, in the lymph node or the spleen. So B cells, which originate and mature in the bone marrow, when a virgin cell is triggered by an antigen and cytokines from the T cells, the two cells are formed. Plasma cells, and I'll just go back here one, okay? Plasma cells are going to be the ones that secrete the antibodies. So plasma cells that mature and produce antibodies specific to a single antigen. This antibody only fits the red gumdrop bacteria, not any other ones, okay, or not any other viruses, okay, to a single antigen. When the fighting is over, they will commit apoptosis in order to maintain what? Homeostasis. Good. And then it will also form memory cells. And the memory cells, right, are cells that will recognize the same antigen in the future. Okay. This production of millions of clones, that's called clonal selection. Clones of the cells that recognize and produce antibodies, they build an army of cells that do the same, All right. So here's your antibody-mediated. So here you can see you have these different B cells and you have these different antigens around it. They're called virgin B cells if they've never had an antigen that bound to their B cell receptor. Now this one has, and it's also receiving cytokines from T cells, right, that have that same experience, but it was presented to the T cells on an antigen presenting cell, and I'll show you that in just a little bit. Here's your clonal expansion, and you're forming both memory cells and plasma cells, and remember it's the plasma cells that generate the antibodies, and when the battle is all done, then they will commit apoptosis. Okay, now I want to remind you about your red blood cells. If you have type A blood, okay, if you have type A blood, then that means you have A antigens on the surface of your blood. Type B is B. Type AB, you have both A antigens and B antigens on the surface of your blood. Type O, 0 antigens. Now, if you have A positive, A negative, what that's relating to is another second antigen called the Rh antigen. So if you are A positive blood, that means you have the A antigen and you have the Rh antigen. If you have O positive blood, you only have the Rh antigen. If you are O negative, you don't have A, you don't have B, and you don't have the Rh, and that makes you perfect candidate to be a universal donor, because you have nothing on the surface of your red blood cells that would act as an antigen to stimulate an immune response. Okay. All right. Now, let's take a greater look at those antibodies and what they look like, okay? So there's 4 chains in an antibody, 2 longs and 2 short. So here are the long chains. They're called the heavy chains, and 2 short chains. Up here is this curve right here is the antigen binding site. Now remember, these are proteins. Remember, proteins are specific to their shape. So this shape will only fit this particular antigen, all right? Now, the bottom right here, the heavy part, okay, that part, there's 5 different categories of antibodies, and different things happen depending on the type of category or where they come from. So that determines what happens to the invader. So remember when I talked to you about exon recombination to form the variety? So when you look at this, it says genetic recombination brings certain DNA segments together. So here you can see this would be like in your DNA or in the mRNA ultimately that you make from it. You can see V1, V2, C, and V3. Okay. And so you can have, depending on what gets clipped out of the messenger RNA, you can have different combinations of genes, which then gives variety for these antibodies, depending on the heavy and the light chain and what their shape is. So on your notes, you have antibodies / immunoglobulins, IG, that's the same thing. Antibodies are immunoglobulins. They are Y-shaped and they bind to and mark antigens for destruction. And the mode of destruction they're going to employ is going to depend, like I said, on one of the 5 classes. I'll give you a picture of that in just a minute. So here, let's look at this B cell right here that's been activated by this microbe. Okay, and if it was a virgin B cell, it would also need to get a signal from a T cell that also was presented with that. So this B cell is producing antibodies. So these antibodies, when it tags on to that antigen, it neutralizes it. Okay, opsonization and phagocytosis of microbes. So when it's got here's the microbe, that little thing with dots all over it, right? Here's an antibody on it. So this phagocyte is going to go and engulf it. Antibodies on a microbe or organism can cause the natural killer cells to kill. And then you can see it can also be used in conjunction with complement proteins. So you can see the C3b here. That's the one. If you're going to know anyone, complement fragments, know complement, think C, and then 3b. It's just different fragments of it that are going to be formed. But what you want to know is that a combination of the complement and the antibody can cause that destruction. Remember where it drills a little hole in and it kind of explodes. But also complement also increases inflammation as well. All right. And the complement and antibody can also cause phagocytosis. So here you can see antibodies. One thing they do is they cause what's called agglutination. And this is where, I think I'm picking the right word, I just pulled that out of my head, where because the antibody is Y-shaped, it has 2 arms, so it can hang on to 2 different microbes at the same time and kind of cluster them together so that you're kind of coordinating, I can't think of the word, sorry, long day. You're kind of clustering them together so that they can be wiped out altogether by some macrophages, let's say. And speaking of a macrophage, here you can see you have a macrophage, and it's eating this bacterium that has antibodies pasted all over it, right? So all of these antibodies right here on this particular bacterium make this macrophage really want to eat it. Do you remember what macrophages started out as? Right? As monocytes, right? It's like macrophage is the Hulk and Bruce Banner is the monocyte. All right, so here are your different categories of antibodies. And let me see, I want to check one thing. Yes, there we go. Okay, so these are your different categories of antibodies. So immunoglobulin G is what you hear most about. Just looks like a regular old antibody, like what I showed you, the Y with the heavy chains and the short chains. You do not need to memorize these. In AP, you used to have to, but you do not anymore. But I want you to see them in case they ask you a question where they ask you to apply it to this. So immunoglobulin M, can you see that there are like 5 around here in a circle? It groups them together. IgA is 2. This is found in your throat. I always remember this because when somebody has to look in your throat and see if your tonsils are swollen, IgA is found there. IgD on immature B cells, telling you when they are mature. And then IgE, I would know this one. This particular immunoglobulin, can you see how it's planted itself in a mast cell? And that will trigger that mast cell to release histamine. Okay, so don't worry about memorizing any of this. I just wanted you to see, and if you needed to, you could pause it and look it over. But Ig is probably the most common one, immunoglobulin G. So this activates the complement proteins and phagocytosis. IgM is found in the blood and it gets clumping, so you can take something out if you need to in a group. This IgA is in your throat and digestive and respiratory tract. IgD found on immature B cells, and then IgE is found on eosinophils and mast cells, and it causes an allergic reaction. It really releases a lot of that histamine. Okay, so here you can see a mast cell. You can see the antibody right here with the antigen in it. And so when the base of that antibody, the Y part or the heavy chain at the bottom of the Y, that will trigger the mast cell to release histamine. So it's IgE. That's the name of this antibody right here. So on your notes, you have C, mode, I'm under 5C, mode of destruction, types of Ig determined by the base of the Y. And the only one you need to fill in is IgE. The base binds to eosinophils and mast cells, arms bind to antigen, triggers histamine release. Triggers histamine release. Good. All right. One last thing I want to throw in there. We'll talk about this at the end as well, but monoclonal antibodies, mono means one. So monoclonal antibodies are made by fusing B cells with cancerous mouse cells and generating specific antibodies to be used in research. So they can customize antibodies to be used in research by taking a B cell and fusing it with a cancerous mouse cell and make the antibodies it wants. So you can see it says to select specific molecules, diagnose for infections. I think I have a picture here at the end. We'll talk about it a little bit more. Okay. So under our adaptive immunity, we've talked about B cells. Now let's talk about T cells and cell-to-cell combat. All right. So the target for your T cells, if you remember, are your own body cells that are infected. Now, T cells can only be activated by another whole eukaryotic cell. They cannot be activated by the virus or the bacterium itself. They have to be activated by a secondary eukaryotic cell. So let's take a look at how it does that. Here's a macrophage, and it's engulfing this pathogen. It's destroying it. And like I told you, antigen-presenting cells will display a portion of that antigen. Okay, so when it does that, it can display it, and there would be a receptor, a virgin T cell receptor, who would then have a receptor only for that particular antigen. And so then you would start doing your clonal selection again, so mitosis and differentiation, and you will make cytotoxic T cells, which cytotoxic, cell toxic, will kill. Okay, you will also make memory cells for next time, and then you will also make what's called helper T cells. Helper T cells help coordinate your immune response. So I'm going to show you a couple pictures of that, but I wanted to give you an overview. So virgin T cells have to be triggered by an antigen-presenting cell, and from that virgin, you'll get 3 types of cells: cytotoxic T cells, helper T cells, and memory cells. So one more than what you got with the B cells, right? So helper T cells, what you want to know about them, they are the major driving force and main regulators of the immune response. That's why HIV is so effective as a virus, because it takes out helper T cells. So these are kind of like the wedding coordinator or the general for an entire army, right? That coordinate your entire immune response. So they play a very, very important role. They're the ones that are going to be activating your B cells, right? So they get in the game as well. And then killer T cells, what their job is just to kill, okay? But they kill those cells that are displaying that particular antigen. So on T cells and cell-mediated immunity, it targets our own infected cells displaying the foreign antigen. T cells originate in the bone marrow and they mature in the thymus gland. When a virgin T cell is triggered by a presentation cell displaying the antigen on an MHC II protein, now I have to go over that with you, it will mature and form three types of cells. Cytotoxic T cells that kill any cell displaying foreign antigen on an MHC1 surface protein. I'm going to talk to you about MHC1s and MHC2s. Helper T cells, which are like the Army general encouraging the response, and memory T cells for next time, So and then you still have clonal selection where you make, millions of each of those so you can respond, and then they commit apoptosis at the end. So let's go back to that MHC1 and MHC2 that I was talking to you about. So these antigen presenting cells, when they display it, oh, here is a target cell, here's some cytotoxic T cells killing, all right? The only way you can talk to a T cell, so here's a T cell, is if you display a part of the antigen. And in a previous class, I talked to you about how like those macrophages and dendritic cells, it's like they cut off the head of whoever they're fighting and they walk around displaying it. What they use to display is either an MHC1 or an MHC2 in order to display it. And this is significant. Now, when you look right here, you can see this T cell and whatever is presented to them. If you display an antigen on an MHC1, you're done, okay? You'll be dead soon. You'll be killed. What that communicates to the T cell is, I'm a body cell and I'm infected. Here, go ahead, kill me. That's what's inside of me. Your cells can't keep secrets. They present everything that's inside of them unless it hides in your DNA, right? As a provirus or a prophage. We'll talk about that later. So anyway, so an MHC1, if you present an antigen on that, you're going to get killed. Who has MHC1s? Almost all of your cells in your body are MHC1s. But if you have MHC2s, then you will present that antigen, and that means, hey, I'm a fighter, specifically, maybe I'm a dendritic cell. I just killed this. Here's a piece of it. Can you help me fight it? Okay, so MHC2, I will help you. If you present an antigen on MHC1, you're done. All right, so if you look, let me go to another slide here. Okay, so you're safe, you're out. Let me show you a couple things here. So here's an infected cell. Here's the red cell. Look at the green the green part is coming out of the infected cell, okay? And it's an MHC1. You can see it just has one arm. What it's presenting is this red triangle. The T cell receptor is in purple right here. That's the T cell receptor docking with the red triangle that is being shown to it by an MHC1. Now notice what's kind of descending out of this T cell right here. Do you see this protein right here that docks and double checks? Are you an MHC1 or are you an MHC2? So it can dock and see, oh, you're an MHC1. This is stabilized by what's called a CD8 protein. I can always remember that because I know this infected cell is going to get 8, right? You can think of it like you're going to get destroyed in some way. Now, over here, you've got a antigen presenting cell. So this is a cell that's been infected by something. Oh, sorry. I apologize. Over here, you have your infected cell. This is a cell that has fought the good fight. It could be a dendritic cell or a macrophage, and it's killed that pathogen. It's holding up the head to say, hey, look what I found. And so the antigen presenting cell, this is its MHC2. See how it's got like 2 legs to it as opposed to this one that has 1. It's presenting the antigen as a red triangle here. The T cell receptors in purple. Notice what it's getting stabilized by. It is getting stabilized by what's called a CD4 protein, which I always remember I'm for you, not against you, okay? Now, pay attention up here to the top. Hang on one second. Okay, sorry, I had to get a quick drink of water. So look up here at the top, okay? And let me get a pointer as well. Okay, so notice this says TC. So this is a cytotoxic T cell. This makes sense that it would have a CD8 and it would bind to any cell that has an MHC1 because it's going to kill it, right? This is a TH. Any idea what this H stands for? Helper, okay? This is a helper T cell and it's stabling with the CD4. I am for you, not against you, because I recognize you are an antigen presenting cell and you're gonna show me what needs to be destroyed. So on your notes, okay, go to, we're under T cells and cell-mediated immunity. MHC proteins, there are two types that display antigens. MHC1 is on almost all cells, and these are cells that can be killed if displaying a foreign antigen as well. And then MHC2s are on APCs, which stands for antigen-presenting cells. Antigen-presenting cells. Now listen to the ones that are antigen-presenting cells. Macrophages, part of your innate immune response. Dendritic cells, part of your innate immune response. And B cells. That's it. Now think about somebody who's missing there. Helper T cells have MHC1s, not MHC2s. And that's why HIV, when it's inside your helper T cells, then it wipes out the helper T cell, the coordinator of your immune response. And that's why people who have AIDS, which is when their T cell count is really low, they start getting all of these other infections in their body because they're not coordinating their immune response. They're not coordinating their immune response. Okay. Now, let me move on here to our next slide. Okay, so let's take a look at how this might play itself out. Okay, so here you have this bone, sorry, let me get this smaller. Here we go. Here you have the stem cell in your bone marrow, and here is a T cell precursor. Remember, where do T cells go? To the thymus to mature. And they can, from that, right, you can ultimately form, once you have a T cell that's stimulated, right? Here's a helper T cell. It's binding to an MHC II antigen presenting cell going, hey, look, we have an infection. So it'll start secreting cytokines to activate other immune cells. Remember, you could also have cytotoxic T cells that end up killing. That's what their job is. And they kill those that have MHC1s, right? It's just not showing you memory cells in this picture. That's the only thing that's missing. But what I want you to see is, look at these T cell precursors. They're supposed to go to the thymus gland to mature. And look, this is somebody who was like in Navy SEAL school and like rung the bell and bailed. And natural killer cells are like, could be a T cell if they had gone to the thymus to mature, but they didn't. So they are killers and they are effective, but they're just not Navy SEALs. You won't have them learning and getting better in their response. They will kill any antibody-coated cells, right? And they will kill anybody displaying an antigen on their MHC1, or they can kill any cancer cell, but they don't have the ability to adapt and learn. They kill anything that is foreign. All right, so let's go here. All right, so big picture. Non-specific defenses, those are part of your innate immune response. Recognize cells or objects as foreign, you work to remove it. That's your whole inflammatory response, natural killer cells, protective proteins, etc. Okay, then your specific defenses, these are your, and I put this on here in case you want to take a screenshot basically of it, but these are your B and T lymphocytes, B and T lymphocytes, and they are synthesized from maybe 1 or 2 virgin cells originally. It could take 10 days to 14 days, but it's the only way you're going to absolutely wipe it out, because you're going to make a clone army that just target that specific antigen. And then memory cells are also made for next time a faster response by both B and T cells. B lymphocytes make antibodies that tag foreign cells for destruction, remember, from their plasma cells that make it. And T lymphocytes destroy our own body cells that are either cancerous or they're harboring some sort of pathogen within them. All right, so let's take a look and apply it here. But I am going to take a quick minute to pause this video, and I will start right here on video 3. All right, good job.